Good morning, everybody, and glad to have everybody here. This is certainly different than what we're used to doing. And I can't say enough about thanking all of y'all to come out here this morning. And so this is a different kind of meeting, and lots of good stuff to share with you today. And the big thing on the presentation I've got kind of a lead in to what we're actually doing and what's been going on with restoring the Ozark Chinquapin. Lots of good stuff. We are blessed, is all I can say. And last night we had our board meeting, and I was just very humbled and honored that we have so many talented, dedicated people who are all over the country making a difference. And one thing, and I see Leslie up here, so one thing I would like to do is kind of borrow a little bit from her. I borrow things people say quite often, and Leslie said something really good that stuck with me years ago. She said, the Ozark Chinquapin tree has never changed. The only thing that has changed is our knowledge of the tree. So today we want everybody to sit back, enjoy yourselves, relax, learn a little bit, ask questions. And so we've got presentations about restoration that we're doing, about black testing that we know that we've got some really good genetics, some even better than the Chinese chestnut, Ania mellissima. And so there's been a lot of work that's gone into this. And if it weren't for all of us pulling together doing this, none of this would be possible. That's just all there is to it. And so it's a great thing. And then of all things, we also have a little bit of music today. We have on our board two members that will be singing a new song that they wrote about the Ozark Chinquapin, and really entertaining. And about every 50 minutes or an hour, give or take, maybe more, maybe less, we're going to give you a break. And then we're going to take a 45-minute lunch break. We think it's real important. It's a real long meeting that we're doing. But for all of us, we work full-time jobs. And for a lot of us, we're taking vacation time again just to do this. And a lot of that is not known by people. So a huge thank you for everybody doing this. And so we're going to talk about several different things. We've been doing a lot of work on RPM trees, and we have Kim Young from Forest Killing Nursery going to share a little bit of information on that. And then we've got Hannah Pilkey who's going to share a little bit of research that she did on the OXO and why the Ozark Chinquapin has resistance, and some interesting things that we have there as well too. And so here we go then. Okay, so my name is Steve Bost, and I'm the president of the Ozark Chinquapin Foundation. I'm just kind of the person that kind of helps pull everything together. It's definitely a group effort. I'm not the boss. I'm just kind of the one that kind of helps organize some of the stuff. And so I want to keep that in mind. So if trees could talk, what would they actually say to us? And so that's my son, Will. He's about, I don't know, 10 or 11 in this picture, something like that. We got a young Ozark Chinquapin there that's, I think, less than 3 years old with nuts on it. And so the good news that I want to share with everyone today is that we have, through years of work, a blight-resistant Ozark Chinquapin tree. But that's not all of it, though. Blight resistance is not enough. And so we also have a lot of other unique traits that we've bred into these trees that we've changed, amplified, and we're going to share this with you. So that is the success story of today. So for over 14 years now, through field and laboratory work, our mission has been to create a blight-resistant Ozark Chinquapin tree. And the other thing is preserve the genetic diversity of the range of this tree. So we're trying to save it, by producing or creating a blight-resistant tree, something that, according to the experts, is not out there. And then the other thing we're trying to do is preserve the genetic diversity. And so this is 100% pure Ozark Chinquapin that we're dealing with. We don't use hybrids. We try to find natural resistance. And so, so the number one thing that we had to do, first of all, years ago, was find surviving Ozark Chinquapin tree, not a stump sprout. We had to find that rare one, and we're up to about 46. Now that 46, we had to remove a few of those from our breeding program because there was some qualities about them that may not have been the best thing for the tree. So we tried to take out those 46, narrow it down to the ones that we thought were the best. So on our search, we started back in 2005, And so if you look to the left, that's a tree in southeast Missouri, and that's a witness survey marker on it, and dead snag still standing. And so we look and look and look, and what you're going to find is just remnants, kind of like almost dinosaur bones of what was once there. And and if we did find any trees, here's what we found. Thousands and thousands of blighted stump sprouts are in the process of it. It's a repeat process, and it's all over the floor. And so the same blight, Cryphonectria parasitica, that wiped out the American chestnut, started in Virginia, and we had Ozark Chinquapins in Virginia, and it was doing the same thing there. And it was spread across the Mississippi River to where we have larger populations here across on the west side of the river. And so this is what we were running into. So our question was, there's got to be some out there. And so I had time and time again, here's what I kept running into when I first started trying to do this. And AJ, we talked about this, and you actually did an article or two, I think, where you talked about the ghost tree that we were looking for, and we were told they did not exist. But from what I knew about history and science and pathogens and diseases, it's extremely rare for disease or pathogen to wipe out 100% of the population. And so the question I have whenever we might find one is this one isolated? Is there hypovirulence taking place? Something else possibly going on why this tree has lived to be as old as it is. So voila, through grassroots, or what I call tree roots efforts, working with farmers, ranchers, landowners, foresters, we have this network of people that are out in the woods, and we're searching and looking, and extremely rarely will we find a tree like this. So I don't want people to think that we went out and this was easy to do. It was very difficult, and I've got somebody going to speak here just a little bit, tell about how difficult it was. And so we began to find these extremely large trees, and what's significant about this is that they were in heavily blighted areas. It was not like there was no blight there, they were isolated, although we did see that a couple of times. And so these trees that we're finding, we began to collect grass from them, pollen from them. And then back in 2006, I began to working with people, and we established at unknown locations research test plots. We have a total of 17 of those now, and those are scattered over mainly Missouri and Arkansas, and in Oklahoma. We have one on Cherokee lands right now. And so these trees, the genetics of them are very important, and several of these trees were around back when the blight swept through. So there's a lot going on here. So we came up with a recovery action plan on how to actually save and restore these trees. And I don't think I have it on this, but yes, I did. The research test plots are a big one. We're dividing those genetics out to different locations where we're doing our research work, our cross-pollination, controlled pollination. And so we collect seed, pollen, grass, and do DNA testing. And so actually knowing that there's no foreign castanea in the genetics is very important. If it is, we don't want it. And so Sandra Anagnosticus, I saw you earlier, and I cannot say how talented this lady is. And so we sent some to her, and she did some hydrogen peroxide testing. And if I remember right, and she might chime in a little bit later here, it will only tell you if there's any foreign castanea in the sample to her. So preserving genetic diversity of the native range, that is huge. And so some trees that are very prone to blight, we save them. And so you never know, there may be a new disease or pathogen come along, and we may actually need those genetics again. Cross-pollinating surviving trees with desirable traits. We did a lot of work on this, and the ones that were actually stood out for us because of different reasons of qualities they possessed, we would, a lot of times asexually reproduce them and to speed up the process, and then cross-pollinate them, controlled pollination on a research test plots. So the only way really to know if that tree is truly resistant to the blight is to inoculate it. later on. Leslie's going to share some things with us on that. Hannah Pilkey is too. Science and genetics and hypovirulence, raise public awareness, a lot of outreach and education, and outreach is huge. We have a lot of people on our board. They're actually interpreters, and for a living, they actually do public programs and raise public awareness about different things conservation minded. So raise money for it. We're probably the least funded organization I know of. We talked about this last night. If you give us $1, that dollar goes directly to it. We have no rent. We have no utilities we pay. There are no salaries. We all are working people that sacrifice unselfishly our time and effort to make this happen. And that goes for our members as well too. And stop advancing. I don't know what I did there, but it stopped advancing for me. Okay, there we go. And so dedicated locations in and out of the blight range. That's important because we weren't for sure how successful we would be. Seed to the Ozark Chinquapin Foundation members for reintroduction. And this is like playing a football game. You put the ball into the hands of the player who can do something. And with us, we've got a lot of very dedicated members that they believe in this tree. They want to restore them back to their property for the benefit of wildlife and ecosystem, and for people to enjoy too. So we expand our restoration now, and we've been doing this for a while. And this year, and last year was a huge year. A lot of state and federal agencies getting on board with us, and so we've gone from putting in research test plots to now where we're doing restoration plots. Coming up just a little bit, Chris White's going to tell us a little bit about one that we put in. So how is resistance possible? The answer is genetic diversity. You have high level of genetic diversity when you have genetic diversity taking place, and resistance is extremely, extremely rare. We're looking at 0.01 to 3%. Very, very, very small percentage. And I try to tell people that when we find these trees. For those of you that hunt, they'd be probably the equivalent to Open Young or Boone and Crockett buck. Exceptionally huge and stands out from the rest of the population. So a lot of genetic concepts and theories and practices, and so natural host resistance is a solution. And we've experimented with lots of different things. We've created graphs and that sort of thing to actually accelerate the process, but we go 100% pure Ozark Chinquapin, no GMOs, no transgenics, no hybrids, but we experiment with a little bit of everything. So we don't close any door on scientific research, and all this work we've been doing allows us to retain genetic diversity. And so it's going on with others. It's tough out there on the forest today. We continue to lose species. So with the increased proliferation of trade and travel, except for this year, there's a lot of unwanted pests and pathogens that hitchhike and make it into this country as well too. And so throughout it, you can see some success stories. Some trees like the American beech that are suffering as well too. They actually, if I remember correctly, about 6% of those actually have some kind of resistance to the beech bark disease. And so those will actually bring the population back. Those have a real high percentage. We weren't dealing with that here. So I like my little cartoons right here. So it's trying to make this simple. So if you can imagine our approach to this, if you have a heavily blighted forest in Missouri, and you got trees that are dying, you have a closed canopy, and basically no nut production. If you do have any nut production, what few nuts there are eaten very quickly because they have higher protein than the other trees. So so that equals a tree that's functionally extinct. And so that's what we're dealing with. So let's imagine this represents an area of several hundred miles. We find one tree. problem you run into quite often is there's no other trees around to actually pollinate it. There's too much space in between. So what we're doing would not happen today because of roads, highways, cities, and dissected forest. You know, the forests we have today are very dissected. Get an airplane, it's really obvious what's going on. So what we're doing would not happen ordinarily. So you look at another place, a heavily blighted forest in Arkansas. Trees dying of blight, closed canopy, no nut production, also functionally extinct. So what we've done is we would find these trees, and keep in mind they're few and far between. Some of these trees may be hundreds of miles apart. What we do is we actually will cross these trees, and we'll take A and J and we'll cross it with controlled pollination if they're ones that we see are what we want. A lot of times to accelerate this, we will maybe collect grafts from them, and on our research spots, we'll collect the pollen. Sometimes I've done it here, and you know, we've been drying and freezing pollen since I guess about 2006. So then you take AJ and you cross it with Q, and you got AJQ. And then you take AJQ and you cross it with another tree with desirable traits. And so then once you get those, you have you know, different crosses and crosses and crosses. And then out of that, roughly 2 out of 10 of the crosses across the board are going to be have less resistance or less of those characteristics you're looking for. 6 out of 10 will have it. 2 out of 10 though will really surprise you. A lot of times they will have greater desired qualities, whether it be nut production or blight resistance or resistance to the blight, than their parent trees. And those are the ones we've been focusing in on. So the results. So what we've got is an Ozark Chinquapin that has high blight resistance, Cryophaenectria parasitica, and it's the ones that we've been testing, and we're going to show you a little bit later on, are as good as, if not better, than Chinese chestnut. But don't get me wrong, some of the testing that we did, we found out we produced some that were even lower resistance. Some that were getting real close to the Native American chestnut. These trees are fast growing, early nut bearing, large nut crop, and September through October. And at one time, we thought that September was the main month to put on nuts. We have several trees in our breeding program that we've crossed. And again, this is where you have genetic diversity, where they're actually having nuts ripen in the month of November. And sure, the squirrels thought it was a squirrel feeding lot, not a research test plot. We had a lot of issues there. So fast growing early nut production, large nut crops, immune to root rot disease. The Phodoptera synonymi is a big one. And you know, it's been here for a while. We have no problem with it. And we'll explain a little bit later on some of the reasons why that is. And so resistant to Asiatic gall wasp. And so Sandra Anagnosticus did some great research, and we're still benefiting from what she did. And she found out that the American chestnut is very susceptible to the Asiatic gall wasp, but the Ozark Chinquapin is not. Real high resistance to it. Let's see, nuts are higher in protein and carbohydrates than white oak, red oak, American chestnut, Dunstan chestnut, Chinese chestnut. And the trees that we've got are best of the best that we've created, are rugged. And once they're established, they can tolerate drought, and they can coexist with the Asiatic oak weevils. And I'm going to show you some pictures here, a little bit of some of the things that are going on. And these trees, very low maintenance. We may water them a little bit. Some of them we don't even water at all. And we've lost some, but the ones that do survive are very, very rugged. Under good conditions, unfertilized, first nuts in 3 to 5 years. And we've been giving a lot of these good genetics out to board members, are members of the Ozark Chinquapin Foundation. Some of them are already getting nuts back and have been for a while. These things are good to eat. I mean, what they lack in size, they make up in protein and carbohydrates. And we don't put everything on here, but some of the trace minerals, they're off the hook, especially the magnesium. So it's a plentiful, high protein, yearly dependable nut crop that benefits natural ecosystems and people. And so here's a few pictures. So ordinarily in Ozark Chinquapin, you'll have one cluster of birds along about a foot back from the end of the branch or so. And typically in the wild, you'll see maybe 2, 3, 1, possibly 5, sometimes maybe 8 or 9, but that's pretty well about it. There's a few exceptions. So now we've got trees like this one. This is 47 nuts on the end of one branch. And so this didn't happen accidentally through selective breeding and crossing of 100% pure trees. We've actually amplified these traits. And so here's another one. Leslie, I didn't tell you about this. I like what Chris White said. He said he and I have faces not presumed are made for radio, and I thought that was pretty good. So on the right is a tree that I actually planted on a research test plot, and this location was not ideal. And I'd say on a rating of 1 to 10, 1 being the worst location and 10 being the best, this is probably about a 5 or 6, somewhere in there. And so this is a 3-year-old tree you're looking at on the right. And here's Leslie. This is in October. Here's that same tree, a little bit less, slightly less, almost 8 inches DBH. And so getting back to some of the things we were talking about. So one thing that that really troubled me at first, I guess, was the fact that we had a lot of predation on these young seedlings that we were planting, or I'm sorry, these nuts that we were planting. We don't plant seedlings. Any nut-grown tree can outgrow any potted tree, and it's because of the taproot. And I've got, not on this one, I got a picture of some of this later. So in a period of about 4 or 5 weeks, you're going to have about a 32 to 36 inch long taproot grow, and that's the key mechanism to its survival. So this is a volunteer tree that came up on one of our research test plots that was not eaten by the deer or the squirrels. And you can see all the holes in this from the Asiatic oak weevils. They love chestnuts. That helps us. Sometimes we try to find them. We just look for one that eats the most. On the left, this is a tree branch I accidentally cut down. And this tree branch was about 31 feet up where I cut it off at, and one of our trees on our research test plots, one of our real good genetics. And this one, if you look at, there's no damage. So why is it from 10 feet down, you're having a lot of high predation on the leaves? And so I couldn't understand that. And so the answer, and I made a video of this, is not a very good one, but here's our hero up here at the top. This is one of our neotropical birds in northern Parula. And they love to go to Ozark Chinquapin research plots or any place they're growing, especially out in the sun, and they will glean and pick off the insects from these leaves. We got a lot of help here. And so that's what's going on. And because of bobcats and mainly gray foxes, if northern Parula or any bird is less than 10 feet, a bobcat can get them. And I've seen enough things on trail cameras to know this happens. So for their survival, they stay up usually about 10 feet or more. And so that's why once the trees get up about that tall, we don't have an issue with them, so they can coexist. So if you bring back something to a healthy environment, and birds like northern Parula, this is just one of the little unknown things of the pages that have been waiting to be filled that we're finding out about, and they play an important role in it. And so these are pretty rugged areas where we plant these trees at. They're not babied at all. So here's a unique graph we put together. This from research, from different sources, credible sources, and AJ Hendershot put this one together. So if you look, the Ozark Chinquapin on the carbohydrates, it's, if I remember correctly, that one study was 61%, just a little over 15% protein. And look how this compares to other castanea or the white oak. And so if you look at the white oak, which I'd always thought was the #1 tree for wildlife in the forest, we're like 3 or 4 times the protein of it. American chestnut, Dr. Fulbright told me 5 to 8% on the protein, and carbohydrates is over 40%. And American chestnut, this is the Dunstan. You can look at this one as well too, and this is right at 10% protein. And the Chinese chestnut. And so that kind of gives you an idea of what we're actually dealing with here. And a short little blurb here along this before I move on. The nursery that we have in the state of Missouri, of Licking, Missouri, years ago. They got like a double handful of Ozark Chinquapin nuts. They were just excited about it. Really rare, our S2 state rating, critically endangered. And they raised 3 or 4 million native trees there again. And the nursery manager told me, so we plant these in our greenhouse, put them in pots, which we're supposed to do. And he said, the next day we saw something I've never seen before. Chipmunks, pack rats at night, possibly, and gray squirrels during the day had shoot holes in the walls of the greenhouse. And when they went in that next morning after planting them, they had gray squirrels running all around the greenhouse. They didn't know why they were there. By the time they figured out what was going on, they had already dug up and eaten every single Ozark Chinquapin nut and left everything else alone. All the walnuts, all the oaks, persimmons, pawpaws, you name it. So this brings to the end of my conclusion. So what we're doing, it's taken an incredible amount of work from our members that we work with that consistently get out there, and they've got jobs, and they sacrifice unselfishly their time and their money and their efforts to help do what we're doing. And it truly is a group tree effort, and something we can all be proud of. So if you're out there, raise up your hand and pat yourself on the back for a job well done. So for the future generations to inherit, it's important for our wildlife to inherit. And that's a little baby bear cub, and they'll eat a little bit of everything. The mama bear will. They're dependent on the nut crop for reproduction. If you don't have it, they will abort all the fetuses. On the right, there you go. They're good to eat. And so with that, that's the end of my presentation.